In this video, we're going to take a look at the process of DNA replication. This process is really important because in order to pass on your genetic information to your offspring, whether it's another cell or an organism, you have to make a copy of your existing DNA. And the process of copying that DNA is called DNA replication. And when we look at our DNA molecules, what you have to remember here is it's a double-stranded DNA molecule that is complementary, which means if you have an A in one strand, you will have the complementary base T in the opposite strand, and likewise, if there's a C in one strand, there's a G in the opposite strand. The other important thing we have to recognize about our DNA molecule is not only do you have complementary strands here, but those strands go in opposite direction in what's known as anti-parallel strands. And if you look at our molecule that is undergoing DNA replication right here, this is from a prokaryotic cell because it's circular. Eukaryotic cells, if you remember, have linear DNA strands. And this process is going to end up doing what is known as semi-conservative replication, where the new daughter DNA molecules that are made are composed of one old strand and one new strand. And so if you look carefully at this process right here, what you can see is the new DNA molecules have one dark strand and one of these lighter purple color strands. So it's semi-conservative because DNA replication results in one old and one new strand in the DNA molecules that are made as a result of this process. And if we look at this process, it starts out in one location on a prokaryotic chromosome and multiple locations on eukaryotic chromosomes. And this is known as the origin of replication, which is basically a short sequence where this process is going to start. And the first step in this process that we see in our cartoon right here is this enzyme that is known as DNA helicase is going to come in here. And its job is to basically unwind that DNA helix. So it uses a little bit of ATP to provide energy to break the hydrogen bonds that are holding the two strands together right there. And that helicase is going to move down the DNA on both sides of the origin of replication. And so you end up with what are known as replication forks here. And both sides of our DNA are opening up to give us those replication forks. If we look at our big picture, and then in this little screen over here, we're just saying what's happening on one side of that origin of replication. And this leads to a problem because when you have those single-stranded DNA molecules floating around, they're really not as stable. DNA really likes to be in this double-stranded configuration over here. And so what the cells do is they coat those single-stranded DNA parts over here with some stabilizing proteins. And that just basically keeps the two strands from sticking back together and forming those hydrogen bonds, and also helps to protect them a little bit so that other enzymes don't go in and start to chew up your DNA, because that's what we do with virus DNA that can be single-stranded and floating around. So we want to get rid of that DNA. And if we look at the next step here, we're going to see an interesting little process go on here. An enzyme is going to come in and knock off some of those DNAs, and it's going to make some short little RNA strands. And so if we look at this process here, what we're looking at is an enzyme that's known as primase. And so you can see it makes short RNA copies of our DNA. Again, these are complementary bases that are forming hydrogen bonds with your existing bases in your DNA strand. Once you've got your RNA strands, another enzyme can come into this process here. And this enzyme is known as DNA polymerase. And you can see what the enzyme does. It basically has the nucleotides will form hydrogen bonds with the existing strand right there. So again, A with T and C with G. And then this enzyme is going to come along and use those existing bases that have lined up and just start attaching them. And that makes our sugar phosphate backbone right here. So the energy for this process actually comes from the hydrolysis of those nucleotide triphosphates. And if we look at our top strand right here, what we see is you make one short RNA primer, and then DNA polymerase just starts adding bases, and you can keep growing that strand in what's known as leading strand synthesis. You make one continuous DNA molecule using your existing strand of DNA as the template. So this existing dark strand right here determines what bases are going to be laid down and connected together to make that complementary strand right there. And so think about this process. Our leading strand is just going to keep following along, but notice your helicase keeps chugging along here and it keeps opening up our DNA strands right there. And the opposite strand, the problem is this is anti-parallel. So if our top strand is going in this direction, the bottom strand is going in the opposite direction. But your DNA polymerase on the leading strand is chugging along, but what about this opposite strand? This one lags behind because your DNA polymerase can only work in the direction to the left side. And the solution that cells use universally is to make what is called an Okazaki fragment after the Japanese scientist who discovered this process. You make your short RNA primer here just like you did on the top strand, and then you add your nucleotide triphosphate bases to that short RNA primer, and now you've got your DNA strand made there. And if you look at this process, you make another RNA primer right there as you open up your DNA helix with the helicase, and then your DNA polymerase can come in here and just fill in this gap. And notice you're left with this piece of RNA primer right here. And so another enzyme can come into this process and basically get rid of that piece of RNA, and then you fill in the gap with your new DNA bases right there. So you're going to end up with what looks like a continuous piece of DNA, but there's one slight problem at the very end of this right here. You can see there's a little gap in your sugar phosphate backbone right there. And there's another enzyme that's going to come in here, and it's going to be able to seal up this gap right here. And this enzyme is known as DNA ligase, and its whole job is to use a little bit of ATP to provide energy to actually seal that sugar phosphate backbone. And once that's done, you've got your completed double-stranded DNA molecule on the top and double-stranded DNA molecule on the bottom right there. So you can see semi-conservative DNA replication is going to result in the creation of two new DNA molecules. Half of it is old DNA strand, and then the other half is some new DNA strands. Hopefully that helps you to understand this process.